So thank you very much for giving me the opportunity to now round up the Munich part, and I will be indeed speaking about genetic and environmental influences. And now I need to search. What do I move? Okay, so we had this theme of favorite slides, starting off slides, and this is actually my favorite one. The WHO revised their strategy for non-communicable diseases, and it was a 4x4 strategy focusing on cardiovascular disease, respiratory disease, cancer, and diabetes, and the major chronic risk factors which were known at the time. However, then in 2018, they added one important disease area, mental health and neurological disease, and that was just discussed as being an important entity to also be monitored, and air pollution, which is an environmental component. So if we move forward and we want to understand and prevent and cure disease, we just had this fantastic presentation of how things are changing over time. And so a lot of the estimates are relying on data of the past, but we have now really a lot of tools in hand. And I will start with genetics, then talk about epigenetics and come to the environment and the outlook. And so we have been mapping the genetic determinants of disease, and the GWAS catalog gives us more and more information. And we have heard a lot about polygenic risk scores already, but I would like to highlight one of the discoveries which were made by colleagues of mine here in Munich, and which are early on, maybe more than 10 years ago, namely linking genetic variants to metabolomics. And what was really transforming at the time was that these linkages between metabolites and single nucleotide polymorphisms were explaining much more than GWASs at the time. And we have seen today very nice examples how this data can be used and integrated. And the genetic information is important because that is more or less, at least is constant in terms of the code in many aspects throughout the life course. So it's with us the entire time. And Mike just said, well, the polygenic risk scores may not work that well. Well, from a cohort, large-scale cohort perspective, actually, it depends what information you have in hand, because this is a recent paper based on UK Biobank, which interrogated the polygenic risk scores in utero exposures to tobacco smoke and active tobacco smoking as a risk factor for lung cancer. And you see on your left-hand side the impact of in utero smoke exposure modified by polygenic risk score, which by itself isn't statistically significant. However, the active smoking and whether somebody started in childhood, which gives the largest cumulative dose, in adolescence, in adulthood, or never smoked, was highly modified by the polygenic risk scores. And if you combine that kind of data and compare those who were not exposed in uterine cycles and who were exposed in squares, you see that there is indeed a powerful interaction across the line. So this is not confounding. I think this is important. These are independent risk factors, and genetic information, the lifestyle information, and move forward with this to really disentangle and understand and be better in preventing and curing disease. And we had a number of very nice talks which kind of were using omics, which you can also from an exposome perspective can call the internal exposome. So variant the results of lifestyle and genetic interactions. And so with that, that brings me to a topic which was not touched on so thoroughly today, namely epigenetics. And The idea is that our lifestyle, our environmental exposures jointly impact or modify the transcribed genetic information, and then we move into disease development. And we were amongst those who very early on identified methylation sites in the whole genome, which were related to body mass index. Interestingly, these not which led to high body mass index, but rather modification within obese persons, which then kind of adjusted the blood cell environment to the obesity. And here, this is now the readout looking at the score of these methylation sites on the development of incident type 2 diabetes. And you see here the estimates in normal people, in overweight people, and in obese people, and for the quantiles of the scores. And this, it's quite clear that, again, here also we have this interaction that in those who are obese and have the highest methylation score, the risk of developing type 2 diabetes is tenfold. And what is really also interesting is when you look at the numbers, because this is not rare. It's equally distributed around the quantiles in the normal people, but here it really accelerates. And so what is important in the pan cohort aspect is that we develop this methylation score in Caucasian-based cohorts, however, then tested it in Indian Asians living in London. And so it really translates across across population groups and ancestries. Another is, so we now moving on, we did similar analyses who did GWASs with all the methylation sites, and were able to discover a lot of trans-regulated CPG SNPs, which then were linked to CPG sites. And again, you start to be able to tell stories and, for example, identify a protein, a gene which encodes for tyrosine phosphate activity, and then is also linked to transcription, is a post-transcription-based biomarker for gestational calorie restriction, and may drive program susceptibility to obesity and other diseases. And then when we went into doing co-localization analyses, these findings were all corroborated, so that you can, for example, identify potential mediators of both genetic and environmental exposures underlying adiposity and cardiometabolic disease. And so that brings me to the last part of my talk, namely the environment. And here we have indeed a changing situation. We have the climate crisis, which changes our environment. We have an aging population, which makes us assume that we have actually more vulnerable populations. We had the corona pandemic, and we saw today really fantastic examples of how that changes health and how we can use omics to understand and detect the most vulnerable people. And then we have an increase in social disparities. This is something which happens globally, but potentially which also is may become a major topic for us in Europe, even more so than before. And so if we now think of environmental exposures, we have this gazillion of potential exposures. Air pollution, pesticides, nanoparticles, chemicals, water pollution. So how do we understand that using novel tools? And one idea we had was to actually think about the molecular impacts, to find that how these different environmental exposures actually could pinch onto major routes to disease development. And we have heard today beautiful examples how this is interrogated doing genetic and other omic technologies. And the other idea we had was to start from concepts of molecular aging. So the idea being that if these should be common pathways, we should actually find them with pathways which had been identified for molecular aging. And again, we had beautiful talks today, kind of using different approaches to characterize aging in more modern terms. But you see here altered intercellular communication, you see in genomic instability, telomere lengths, the epigenetic changes, and the mitochondrial dysfunction. And so we kind of went ahead and conceptualized how we can think about environmental exposures. And we see similar things like in aging, such as oxidative stress, which has been studied a lot. And going back to the Asian presentation, we even saw that oxidative stress plays an important role in space. We see genomic alterations and mutations. And here I would like to highlight an important piece we saw in the paper, namely that environmental exposures can also impact non-coding regions, and that the whole genome sequencing data, which is now coming about, may not only discover genes for rare diseases, but it may also discover kind of very somatic variations in the genome, which we have underappreciated so far, and which in experimental settings can be shown to be linked to environmental exposures. We see the epigenetics, we see the mitochondrial dysfunction, endocrine disruption, altered intercellular communication, altered microbiome communities, and impaired nervous system function. And so one of the approaches we are now taking, for example, using such a concept in a large European project, which is coordinated out of the University of Utrecht, where we bring together administrative cohorts, adults cohorts, mature birth cohorts, and urban labs, where we do the monitoring, which was just highlighted as being so important. And the question we want to answer is how to maximize one's health in a modern urban environment. And metabolomics will play a major crucial role. And we have later in this Pan-Omics Symposium, Gary Miller be presenting his ideas with whom we collaborate on this project. What I also find important is that cohorts allow, of course, prospective analyses, but cohorts also allow to conceptualize causal experiments. So to find people to ask causal questions that within similar people, what would happen if more or less the same group of people would stop smoking, for example? Can this be an experiment? And cohorts can actually be used to turn observational data by pair matching into pseudo-randomized controlled trials, which then, when there is the need to develop test statistics. And this example now comes from microbiome research. But I think it's also important to construct more experiments out of these large-scale cohorts, because you need indeed the large scales to be able to come up with these experiments. And so this is the EXPANS work is part of the European Human Exposome Network, where a total of 9 major research projects are funded to study the health impacts of environmental exposures jointly. And it's the world's largest network of projects studying the health impacts of environmental exposures, that we want to enact a paradigm shift of new systematic understanding of the cause of disease, bringing in novel exposures, using the omics toolboxes, which we have available, and then to create an exposome toolbox, because the environment is largely not considered in many efforts. And these can be solid and cost-effective preventive actions and policies. And so to round up my environmental section, I would also highlight the issue of climate change. Germany is one of the countries, even though we have generally a temperate climate, we are experiencing actually quite substantial increases of temperature. And when you look from the 1880s to today, we have a net increase of 2.5 degrees Celsius during that period of time. And we also in Germany are amongst those countries which have the highest heat-related mortality. We are well in line with Asian countries or the United States. And so this is really a pressing health issue. And we have now modeled the temperature surface of Germany to link it then to our big cohort studies. As Henry has pointed out, we are the cohort country. And what is really telling is if you look at the map of heat days in 2001, we see that there weren't so many heat days. Then we look to a year such as 2015, we see that heat is really accumulating. And it's not everywhere the same. And 2015 is actually not our record high year. It is only the 5th hottest year since recording. And so it's again important to of course do traditional analyses within the cohort, but to also use the omics layers to look at, for example, aging, hallmarks of aging, such as telomere length. And we were able to show that in these hot periods within, this is now Cora data, the telomere length is decreasing. And again, we need repeated measurements, both over longer time periods to look at trajectories, but also within individuals to really pin that down and show the changes already occurring today. And so with that, I come to the outlook. We have these global challenges for health. And my personal opinion is that cohort studies are the ideal tools in a rapidly changing world because we can characterize exposures. We need omics to characterize the genetic underpinning, but also to be mirroring the changes and integrate across diseases and pathways. We need excellent phenotyping, be it variables or be it imaging, and we saw beautiful examples for that today. And we need the diversity of the on the global scale, because the environments and the lifestyles are changing at different rates across the globe. And we are also mixed societies. We are getting more global with people moving around. And so I think we really need to work together to achieve that. From the German side, the NACO health study is an important tool. We have set it. Henry's transmission was kind of not working so well. We have repeated imaging. We have record linkage. Sample collection, which is there to be harvested and turned into data as the UK Biobank. So I think there are tremendous complementary opportunities here where we as the German community can contribute to with the narco health study. And so my last sentence, my take-home message is cohort studies address the challenges of tomorrow, and I thank you for your attention.
Thank you very much, Annette, for your fantastic presentation. Thank you for put a shadow or focus also on exposome. I'm sure we will learn more about in the American part of the session. Thank you for making the broad span from epigenetics, which is also very important to consider in the cohort studies. Please join me in thanking of all speakers, not only in Europe, but also in the morning session in Asia, in Japan, of course, in Europe, in Munich, and the first talks which we have heard today from Boston, US, and the high impact. And I think it was a very good overview and a very success meeting until now. And I would like to hand over to you, Annette, for your summary and closing. Present to you. Thank you.
So thank you very much. It's quite a challenge after such an intense day to summarize the content, and I would do not good service to all the excellent speakers to really summarize. And I've, during my talk, during my keynote, highlighted a few. What is my take home? My take home is that I was so struck with the different angles, how people can look at omics in pan-cohort studies. We have these very large scale cohorts, which produce immense amount of data. And then we have these small cohorts, variables, which also within an individual produce immense amount of data. And these seem to be really different spaces. And I think the way forward is conceptually, but also data-wise, integrate these the strengths of both approaches and think how we come from the narrative of the individual to the population, and where do we need to intervene on the individual level, but where do we need also joint political action, such as, for example, with climate change. Thank you very much.